one, which indicates that the darker one is more concentrated and the lighter one is lesser concentrated, which means as the color of the solution depends, the concentration of the substance present in the sample also increases. This is the basic principle for spectrophotometry. So, the next principle is that every substance has a capacity to absorb to emit certain wavelength of radiant energy. This absorption and transmission of certain wavelength of radiant energy is the characteristic for every substance. For example, if you take chlorophyll, this chlorophyll absorbs only red and violet light, while it transmits or emits only green, blue and yellow wavelengths. This absorption is the characteristic for every substance. Now coming to the overall description, overall definition of spectroscopy is that spectroscopy is the measurement and interpretation of electromagnetic radiation which emits or transmits only when the molecule or atom or ion of a sample moves from one energy state to the another energy state. This is the principle for spectroscopy. Coming to the basing upon the choice of wavelength, it is of two types. The first one is colorimetry and the next one is UV-Vis spectroscopy. Coming to colorimetry, in colorimetry, it is mainly concerned with the absorption of visible radiation whose wavelength ranges from 400 to 800 nanometers. Dear friends, there is an interesting character in this colorimetry is that only the colored substances are get absorbed under this radiation. While coming to the or while entering into the UV spectroscopy, UV spectroscopy means ultraviolet visible spectroscopy. Means its wavelength ranges from 200 to 800 nanometers. In this only the visible light as well as the ultraviolet radiations are get absorbed. Here friends, as I earlier said that only the colored substances are get absorbed under visible radiation. What about the colorless substances? Here the colorless substances are get absorbed under UV radiation which ranges from 200 to 400 nanometers. Coming to the next slide, dear friends, do you know on which principle this spectroscopy works? Here this spectroscopy works mainly upon the principle of Beers-Lambert law. Beers-Lambert law states that the amount of light absorbed is directly proportional to the concentration of the substance present in the sample and also with the thickness of the absorbing material. Coming to the next slide. Here this instrumentation mainly depends upon the 7 components. The first one is source of light, the second one is collimating lens, the third is monochromators, the fourth one is focusing lens and the fifth one is sample holders, the sixth one is photoelectric detectors and the seventh one is digital display of meter. When the light from the light source falls onto the collimating lens, this collimating lens collimates the light to fall onto the dispersion device, that is monochromator. When the light falls on the dispersion phase, that is monochromator, it resolves the polychromatic light into the monochromatic light, or it resolves the polychromatic light into the individual wavelengths and also isolates these individual wavelengths into the very narrow bands, and the selected wavelength is made to focus onto the sample holders. The sample holders are otherwise known as qubits, and the transmitted light is detected by the detectors, usually photocells, phototubes and photomultipliers, and the reading is given out by the digital display of meter. Coming to the source of light, basically sources of light is of two types. The first one is source of ultraviolet radiation and the source of visible radiation. In source of ultraviolet radiation, hydrogen lamps and deuterium lamps are commonly used. In source of visible radiation, only the tungsten filament lamps are commonly used. Coming to the filters, this filter is used to resolve the ultraviolet radiation into very wide narrow bandwidths about 40 centimeters. While coming to the monochromators, this monochromators resolves the polychromatic light into the monochromatic light, or usually in the individual wavelengths, and also isolates these individual wavelengths into very narrow bands. The next one is here basically the monochromators are of two types. The first one is prism and the next one is grating. When the light is made to focus onto the monochromatic device, that is prism, it resolves the polychromatic light into the monochromatic light and usually exhibits this monochromatic light in Bibsia form. The next, coming to gratings. The surface of the gratings is etched. Here when the light from the light source hits onto the sharp edge surface, it divides the components of light into individual components or individual wavelengths. The next one is sample holders. Dear friends, while coming to the sample holders, as we earlier said that sample holders are usually known as cuvettes. These cuvettes are generally made up of quartz. Here this is the cuvette. By placing the sample into this cuvette solution, we can observe the radiation of which the substances are going to observe. The next one is this is the cuvette. While coming to the basic instrumentation on which this spectroscopy deals is, this is the instrumentation for UV spectroscopy or visible spectroscopy. Here we must select or we must enter the required wavelength on which this spectroscopy works and then place this cuvette which contains a sample solution into the respectable place provided in this spectroscopy and then close the lid. Here in this, when the light passes on to the sample holder, the substances which are present in the sample holder, only a few substances absorb this visible radiation. Here the sample holder, here the sample solution which we are keeping. Here the sample solution which we are keeping is to detect the how much concentration or the type of concentration in this solution holds. Here when the light is generally passed to the sample holders, the substances present in the sample holders, only the few substances get absorb this radiation, whereas the non-absorbing substances transmit this wavelength into the detectors, which is usually detected by the detectors, and the reading is given out by the ammeters or potentiometers or potentiometric detectors. Coming to the next slide. Here, as I earlier said that there are three kinds of detectors. The first one is photocells, the second one is phototubes, the third one is photomultipliers, and the reading is detected by the amplifiers, ammeters or potentiometric detectors or potentiometers. The next one is, dear friends, you may have a doubt that or you may have a question. Up to now we have studied about the principles of which this spectroscopy works. Then what for it is useful and how can we use it in our daily life? So, while coming to the applications, here primarily quantitative analysis of proteins, carbohydrates, fats and also nucleic acids. It is mostly or generally used in microbiological labs to detect or to know how much growth the microorganisms are being takes place, or it is useful for measuring the growth of microorganisms, usually bacteria, and it is also used in pharmaceutical industries, food and paint sciences and so on. It is also used in disease diagnosis, pathological estates also. It is also quantitative to determine the quantitative assay of enzyme activities. Dear friends, up to now we have completed the topic, that is spectroscopy. The next instrument I would like to deal is centrifuge. This centrifuge mainly works on the principle of sedimentation. Here this rate of sedimentation mainly depends upon the applied centrifugal field, density, viscosity of the particle and radius of the particle. Here types of centrifugation. Basing upon the types of centrifugation, there are two types. The first one is preparative centrifugation and the next one is analytical centrifugation. Coming to preparative centrifugation, this centrifugation or this technique is mainly concerned to study the separation, isolation and purification of subcellular components like cell organelles, mitochondria, lysosomes, ribosomes, nucleic acids and so on. Coming to the analytical centrifugation, it is mainly concerned to study the micromolecular structure or molecular structure. The next. Here, the outline classification for centrifuge is, centrifuges mainly or basically divided into two types. The first one is preparative centrifuge and the next one is analytical centrifuge. Basing upon the speed of centrifuge or basing upon the speed of rotors, it is differentiated into three types. The first one is desktop centrifuge and the second one is high speed centrifuge. The third one is ultra centrifuge. Again, this ultra centrifuge is differentiated into differential centrifugation and density gradient centrifugation. Coming to the next slide, here differential centrifugation. In differential centrifugation, mainly the differentiation is mainly based upon the size of the particle and density of the particle. As we well known that larger density particles or larger size particles will experience more centrifugal force than the smaller or lighter particles. Here this is the here in this we can in this centrifuge test tube holder, the main thing is centrifuge. The main thing is we can centrifuge cell homogeneous. By applying different centrifugal speeds, we can differentiate the cell components like whole cells, nuclei, mitochondria, lysosomes, ribosomes and so on. By applying different centrifugal speeds is the main important thing. In the test tube or in the centrifuge tube, the cell homogeneous is usually taken. By applying the low speed centrifugation, we can differentiate only the whole cells, nuclei and cytoskeletons, usually in the form of pellets. Then the supernatant or the filtrate is transferred to the another fresh test tube, and by applying medium speed centrifugation, we can get mitochondrial lysosomes, and then this filtrate is shifted to the another test tube. By applying high speed centrifugation, we can get microsomes and small vesicles, and finally my dear friends, by applying very high speed, we can get ribosomes, large macromolecules and so on. So, in this only the pellets are usually formed, these are which are the cell components. The next one is density gradient centrifugation. Dear friends, in density gradient centrifugation, homogeneous gradient and also beyond densities are mainly taken. Basically, there are two methods of density gradient centrifugation. The first one is rate zonal centrifugation and the second one is isopechnic centrifugation. Coming to the rate zonal centrifugation, this entire centrifugation technique mainly based upon the size of the particle but not the densities. Here usually the cell components are separated in the form of a discrete zones. The next one is isopechnic centrifugation. Here the separation mainly depends upon the beyond densities. The next one is types of rotors. Here dear friends, types of rotors are basically two types. The first one is fixed angle rotors and the second one is swinging bucket rotors. In fixed angle rotors, the angle of the centrifuge is fixed to a constant. While the speed of the rotors increases or the speed of the rotor increases, the angle of the centrifuge does not change at all while increasing its speed also. Here mainly the pellets are formed. So, it is mainly used for the cell homogeneous and also it is mainly used for the differential centrifugation. The next one is swinging bucket rotor. In swinging bucket rotor, as the speed of the rotor increases, the angle of the centrifuge tube also changes. Here this is the clinical centrifuge technique. This is the clinical centrifuge. Usually we can see a spinning bucket rotor. Here the angle of the centrifuge changes basing upon the speed of the rotor. Mainly based upon this, clinical centrifuges are commonly seen in clinical laboratories for the differentiation of cell components or cell count, like how much RBC are present in our blood and how much WBC are present in our blood. The next one is applications. Dear friends, up to now we have discussed about the principles and different methods of centrifugation technique. It is mainly used to separate the cell homogeneates, that is cell count, how many WBCs are present and how many RBCs are present, how many blood cells are present in our blood. Here this red journal centrifugation is mainly used to estimate the proteins synthesis, and also it is used for the serum separation and also used for the blood test also. So, this is the basic principles and this is the main concept of which this centrifugation follows. Thank you for giving this opportunity.